Hi, welcome to this session for biology exam build-up preparation as well as revision. In this tutorial, we're simply going to consider biology paper 2 theory for the 2025 GCE exam that was written on 29th July 2025. In this video, we're only going to get to discuss the first 5 questions of section A, where we're going to fully anticipate the concepts needed for a proper exam preparation that can prevent a poor performance. And welcome to this platform for Sensor Academy, either the YouTube channel, the Facebook page, or the TikTok account. Ensure you subscribe to this YouTube channel, turn on the notification bell to get notified when next I post a new video, and tag those that can find this same video very much beneficial. Let's dive in. Moving on to question 1, it says figure 1 shows a palisade cell observed under a light microscope. So in this regard, we're going to find that we've got a cell membrane. This is clearly labeled, and then we've got the nucleus and the cytoplasm. Remember, there is the keyword under a light microscope. Then we've got part labeled A that points to the outer structure of the cell. We've got part labeled B that is beyond the cytoplasm. Then we've got part labeled C, which is found that is in the cytoplasm. So identify, that is, moving on to A, identify the parts labeled A, B, and C in figure 1. So answering this, we're going to find that A, being the outer structure, is going to be a cell wall. So this one is going to be a cell wall. Then B is going to be a vacuole. And as you are watching this video and trying to prepare adequately for an exam, being a student that takes biology to be compulsory, I want you to understand that it's important that you prioritize correct usage of spellings, as this makes your work more correct and attracts the marker's interest in seeing that deservedly this is a clear distinction on merit. Let's continue. Then for C, this is going to be chloroplasts. So A is a cell, B is a vacuole, C is a chloroplasts. Then moving on to, that is B. B says explain the functions of the part labeled A in figure 1. So we're going to say it protects the inner contents of the cell from mechanical damage. So you can say it protects the inner contents of the cell, of the cell from mechanical damage. Then we move on to C. C says give 2 differences between the cell in figure 1 and the one found in the human trachea. Let's say, for instance, the cell in figure 1 is a plant cell, and the cell in a human trachea is an animal cell. So we're going to say the cell in figure 1 is a plant cell, while the cell in a human trachea is an animal cell. Then the second one, we're going to say the cell in figure 1 has a cell wall, while the cell in a human trachea has no cell wall. So the cell in figure 1 has a cell wall, while the cell in a human trachea has no cell wall. And also, the cell in figure 1 has chloroplasts, while the cell in a human trachea has no chloroplasts. Then the last one, state the names of N2 organelles that would be seen if the cell in figure 1 was observed under an electron microscope. So in this regard, when we talk about organelles, these are just simply cell parts. So we can talk about ribosomes. This is where protein synthesis takes place. We can also talk about the mitochondrion, which is a powerhouse hosting respiration. We can talk about Golgi bodies. We can talk about, lastly, the endoplasmic reticulum, either rough or smooth. So here, of course, we're going to say the ribosomes, and then we're going to say the mitochondrion. The ribosomes as well as the mitochondrion. Moving on to question 2. Figure 2 shows the stages involved when testing the presence of a food nutrient W in crushed onion. So then we've got a diagram where we've got warm water and crushed onion being mixed. And then we've got reagent X, which has its drops being added to this existing test tube where there is crushed onion. And then we've got stage 3 where heat is being applied. And of course, the result shows that a ready PPT or precipitate is observed. So it says identify reagent X. Remember, there are basically many food tests in the lab. To mention but a few, we're going to talk about testing for starch. If at all, this reagent X should be iodine solution, then turns out the result would have been blue, black, or brown. And that would have meant we were testing for starch. So reagent X would have been iodine. If reagent X was burette, the result here would have been a purple color or light blue. Now that it is already precipitate, believe me, reagent X is supposed to be Benedict because this is a reducing sugar test. So we're going to say Benedict's solution. Then we move on to B. Explain why the onion was crushed and shaken with warm water. Remember, using the solid parts of onion and trying to test in order to come up with the desired results would have been impossible. But it was better to crush it and shake it with warm water so as to break and open the cells for an easy mixture and obtain desired results. So as to break and open the cells for an easy mixture and obtain desired results. Then moving on to C, from the result in stage 4, which nutrient in the onion was being tested for? In as much as we've said we're using Benedict solution, we are testing for reducing sugar. So this one is supposed to be reducing sugar. Then 2, name the class to which the nutrient identified in C1 belongs. Reducing sugar belongs to a class of nutrients called carbohydrates. So carbohydrates. Then on C, it says name another class of nutrients in onion other than the one identified in C. So onion is rich in carbohydrates, proteins, as well as vitamins. So of course we can just say proteins. Then D, explain the importance of food nutrient W in the human body. Since this is a carbohydrate, we're going to say it provides the body with energy and allows the body to function properly. So it provides the body with energy and allows the body or makes the body to function properly. Then question 3, It says figure 3 represents 3 different models of gaseous exchange in different animals. So here we've got that is the entry of oxygen that is getting dissolved in the blood vessels being the capillary. And then here we can observe the movement of that is the water or gases and then going on to different chambers. Then here we're of course having a direct gaseous exchange that goes to the tissues. So which model represents gaseous exchange in human beings? So because of this blood capillary, we're going to say model 1. So model 1. Then moving on to 2, insects, we're going to say model 3. And then the question continues, give reasons for your answer in A by referring only to the models in figure 3. So we said for human beings it's model 1 because of the blood capillary So because of the blood capillary showing gases dissolving into it, then for insects we're going to say this is because of the direct gaseous exchange to tissues. So this is because of the direct gaseous exchange to the tissues. Then moving on C, it says, which model in figure 3 uses water as a medium of transport for oxygen to the respiratory surface? Of course, we've remained with one model, which is model 2. And then 2, name one living organism which uses the model identified in C1. So of course, we're going to say fish. Then lastly, suggest structures that aid or help movement of air into out of model 1. Model 1 being a gaseous exchange type in humans, we're of course going to have the diaphragm. So we're going to have the diaphragm, and then we can talk about the rib muscles or intercostal muscles. So we're going to talk about the diaphragm, and of course we're going to talk about the intercostal muscle. Then the last one talks about what aids the movement into and out of model 3. Model 3 was said to be for insects. So for insects, we're trying to find that air enters the body through spiracles and then moves inside through a system of trachea. Trachea just branching air tubes. So of course, we're going to say spiracles. And then we're going to talk about the trachea. Then question 4 says figure 4 is a bar chart showing the volumes of urine collected from a person before and after drinking 1,500 centimeters cubed of water. The person's urine was collected immediately before the water was drunk, as indicated at time 0. Then at 30-minute intervals for 4 hours. Then, of course, this is the diagram. So we've got the time intervals, that is in the x-axis. Then we've got the volume of, that is urine. Then we've got, that is the on A, it says, explain why there was a high volume of urine after 60 minutes. Of course, we can say after drinking water, the blood had too much water, which became a dilute. So we can say this is because the blood had too much water, which became a dilute. And then moving on to Roman numeral 2, it says State the point at which the volume of urine stabilized. So if we can observe on the diagram, so we've got 210 and 240 which are standing at the same height, and then corresponding, that is, to almost 100 here. So in this regard, state the point, so we can say 210 minutes. Then we've got, that is on B. On B it says name the organ from which the urine was being excreted. Urine being a component that contains excess water, excess salts, as well as urea, is going to relate to the kidney. So for this one, we just say kidney. Then what process is being demonstrated in figure 4? As long as this talks about how water levels can be maintained in the body, then it calls for osmoregulation. So osmoregulation. Then C, explain two other processes by which water drunk would be lost from the body. So water can be lost from the body through sweating. Apart from sweating, we can talk about through exhalation of water vapor. Then which conditions would increase the loss of water that is from the body? So what would probably increase the loss of water from the body can be a high temperature. Apart from a high temperature, we're of course going to have, that is, a physical activity, which can be in form of an exercise. So that was for question 4. Then we move on to question 5. Question 5 says a man who had normal vision married a woman also with normal vision. They had a color-blind son. Color blindness is sex-linked. Using alleles X to the power capital B and X to the power small letter b, predict the genotypes of the man and his wife. Since we are using chromosomes, there is never a man who is a carrier for a disease, especially which is sex-linked. It's either infected or normal. Now that they've described a man being with a normal vision, we're of course going to use X with capital B and a Y. So this is going to be the correct genotype. And then for the wife, so the chromosomes for the gender of being female is X and X. And now that there is a problem in the discussion being able to have a colorblind sign, this shows that the woman was a carrier. So this is going to be X to the power B, then X to the power small letter b. Then B, draw a genetic diagram to show how this was possible. So in this regard, we're firstly going to talk about, that is, phenotypes of parent 1. So phenotypes of parent 1, this is going to be the descriptions of the two people being talked about. So we'll start with a man. So a man, we can say, is normal. We show mating. And then for the woman, we can of course try to talk about her being a carrier. Then from this, we're of course going to talk about genotypes. So genotypes of parent 1. So for normal, we're going to say X to the power B, then Y. For carrier, X to the power B, then X to the power small letter b. Then from there, we're of course going to write gametes. So for gametes, we're going to say X to the power B. We can circle if we want. We leave a reasonable space. We write Y. We can also circle. So X to the power B, we circle. And then X to the power small letter b, we also circle. This is in readiness for crossing. Then from this stage, we're of course going to cross. So the first one is simply going to come here. So this is going to be X to the power B, then X to the power B. Then the second crossing will be displayed here. So X to the power B, then X to the power B. Then we're going to move on to this one, which is going to be X to the power B, then Y. Then this one, of course, X to the power B, and then we're going to have a Y. So these are going to be called genotypes. So these are going to be genotypes of the first generation. So you can put up that is fertilization here. Then we're going to look at the possible phenotypes. So capital B, this means normal, but remember this is a female child. Then X and X female, but B and small letter b, this one is a carrier, and then this one is normal, and then this one is infected. So this is the color blind son that was described in the question. So this means we are correct, and then we move on. Moving on to C, it says one of the causes of color blindness is mutation. So first one, define mutation. So when we talk about mutation, we can say this is a sudden change or a random change in the genetic material of a gene that is inherited. So we can say this is a sudden change in the genetic material of a gene that is inherited. A sudden change in the genetic material of a gene that is inherited. Then explain one cause of mutation. Mutation can be caused by radiation. It can be caused by a virus replicating the DNA structure. It can be caused by harmful chemicals. So here we can just say harmful chemicals. I hope this video helps to boost the current confidence for the exams. Please leave a comment, share the video, support this YouTube channel as well as Facebook page as well as our TikTok account. Till in the next session. For now, it's a very goodbye.